Hello dear learners, welcome to APCCA LMS portal. I am K. Sai Sudha, LMS presenter, working as lecturer in biotechnology at DK Government College for Women, Nellore. In this video, we are going to discuss on the topic Introduction to DNA Replication. Before going into the topic, let us see the learning objectives. By the end of the video, the student will be able to understand the importance of DNA replication. He would be able to elaborate the basic properties of DNA replication. He would explain the three modes of DNA replication, and he would understand the Michelson and Stahl experiment in detail. As we all know that DNA is the basis of inheritance, it carries all the genetic information of an organism. DNA replication is a fundamental process for transferring genetic information from one generation to another. This DNA replication occurs in all living organisms, and it is the most essential step in inheritance. It is a process of making faithful copies of themselves, that is, the two daughter DNA molecules which are formed at the end of the DNA replication would be exactly identical to that of the parent. This is called as faithful copying of the DNA. Replication occurs before the cell undergoes cell division, that is, during the interphase of the cell cycle. As we all know that the cell cycle is divided into four stages: G1, S, G2 and M. The DNA replication is carried out in S phase or the synthetic phase of the cell cycle. Before going into the details of the replication, let us see some of the basics of DNA. Watson and Crick proposed the double helical structure of the DNA in 1953, for which they have got Nobel Prize. DNA, deoxyribonucleic acid, is a double-stranded molecule which looks in the form of a twisted ladder. The two strands of the DNA runs in anti-parallel direction. If one strand is in 5' to 3' direction, the other strand is in opposite direction. The two strands are connected with hydrogen bonds. So you can see the two strands have hydrogen bonds in the middle, as in the case of steps in a ladder. The components of the DNA are nitrogenous bases, deoxyribose sugar, and phosphate groups. And the DNA is a polymer of nucleotides. A nucleotide is a complex of phosphate, sugar, and nitrogenous base. If only sugar and base is present, it is called as nucleoside. See, this is a nitrogenous base and this is the sugar. Both of them together is called as a nucleoside. And if there is a phosphate, it is called as a nucleotide or nucleoside monophosphate. If there are two phosphates, it is called as nucleoside diphosphate. If there are three phosphates, it is called as nucleoside triphosphate. For example, if the nitrogenous base is adenine, then the nucleoside is called as adenosine. And if there is one phosphate, it is called as adenosine monophosphate. And if there are two phosphates, it is called as adenosine diphosphate. And if there are three phosphates, it is called as adenosine triphosphate. This is the same with all types of nitrogenous bases. Coming to the nitrogenous bases, the nitrogenous bases in DNA or RNA are purines and pyrimidines. Purines are of two types. They are adenine and guanine, whereas pyrimidines are three types: cytosine, uracil, and thymine. But cytosine and thymine are the only ones which are present in the DNA, whereas uracil is a component of RNA. The nitrogenous bases have a capacity of base pairing with each other. The base pairing between purines and pyrimidines is the most important aspect and DNA of the DNA structure and the sequence. The adenine will base pair with thymine with 2 hydrogen bonds. These are the 2 hydrogen bonds. In the same way, guanine pairs with cytosine with 3 hydrogen bonds. These are the 3 hydrogen bonds. So, these nitrogenous bases are present in the interior of the DNA, whereas the backbone of the DNA strand has deoxyribose sugar and phosphate groups. So, the deoxyribose sugars are interlinked with this phosphodiester bond. This is the phosphodiester bond. The formation of this phosphodiester bond is catalyzed by DNA polymerase, which is the basic reaction that is taking place in the DNA replication. So, we'll look into the formation of phosphodiester bond. The phosphodiester bond is formed between the 3' OH group of growing DNA strand and alpha phosphate of incoming nucleotide. This is the alpha phosphate, this is the beta phosphate, and this is the gamma phosphate. So the 3' OH group, which is present on the growing DNA strand, will act as a nucleophile and it will attack on the alpha phosphate of the incoming nucleotide. Then a water molecule is released and an ester bond is formed, which thus forms a phosphodiester bond. Here, the breakage occurs here, and these beta and gamma phosphates are released in the form of pyrophosphate. Pyrophosphate is an energy-rich compound. This is the basic reaction which takes place in DNA synthesis, and it is catalyzed by DNA polymerase enzyme. As we have seen, DNA replication is a very essential process in a cell, and DNA replication follows certain basic rules which are always followed in any condition, and it will be never deviated. Some of the rules are: the DNA replication is carried out by DNA polymerase enzyme. The next one is DNA synthesis always takes place in 5' to 3' direction. So there is a directionality of DNA synthesis. The next one is the DNA replication occurs in semi-conservative mode. That is, the daughter DNA is formed with will have one parent DNA and one newly synthesized DNA. The DNA replication fork moves in bidirectionally. That is, the DNA replication starts at a particular site and the replication proceeds in both directions. That is bidirectionality. And it takes place in semi-discontinuous mode. This is the DNA replication on one template takes place continuously, and on another template it takes place in discontinuous mode. So it is called as semi-discontinuous mode. And the last important one is DNA replication occurs with high fidelity, that is with high accuracy. The error rate of DNA replication is very minimal. So these are some of the basic rules which are followed in DNA replication. So let us see the experimental proof for semi-conservative mode of replication. In 1953, Watson and Crick has proposed the double helical structure of the DNA, and later they have hypothesized semi-conservative mode of DNA replication. In 1958, Messelson and Franklin Stahl has experimentally proved that the semi-conservative mode of DNA replication occurs in a cell. Before going into the experiment done by Messelson and Stahl, it is very important to know the different modes of DNA replication. There were 3 modes of DNA replication that were proposed or hypothesized at that time. They are conservative, semi-conservative, and dispersive. The conservative mode of DNA replication is where the parent DNA is completely conserved in one of the daughter DNA. So, one daughter DNA have both strands are from parent DNA and another daughter DNA have both new strands. Since the parent DNA is completely conserved in one daughter DNA, it is called as conservative mode. In semi-conservative mode, the parent DNA will be divided and each strand will act as a template for the synthesis of new strand. So the daughter DNAs will have one old strand and one new strand. Since only one strand is conserved in the daughter DNAs, it is called as semi-conservative mode. Coming to the dispersive, in the dispersive mode, the parent DNA is fragmented into small pieces or fragments, and the new DNA is formed. And later in the daughter DNA molecule, the parental DNA and the daughter DNAs, new DNAs, are intermixed. So the daughter DNA have old strands, old fragments, and new fragments. So it is a mixture of both old and new fragments in the same DNA. So these are the three important modes of DNA replication. For determining or for proving the correct mode of DNA replication, Messelson and Stahl have done an experiment, and this experiment is called as the most beautiful experiment in biology, or it is one of the classic experiments. For their experiment, they have considered nitrogen, because nitrogen is most important element in the cell and in the DNA, where nitrogenous bases are very common. So he has taken two isotopes of nitrogen. One is natural or light isotope with N14, and the other one is heavy isotope is N15, but this is non-radioactive. It is a heavy isotope. It is slightly heavier than the natural isotope. To distinguish between these two isotopes containing DNA, Michelson and Stahl has used a technique called as cesium chloride density gradient centrifugation. For their experiment, they have taken E. coli bacteria, and they have grown this E. coli bacteria in N15 containing medium for several generations. So this is the experiment they have done. This is E. coli, which is grown in on N15 medium for several generations. So when this E. coli is continuously grown on N15 media, all the nitrogen in that E. coli will be replaced with N15. So when such cells were isolated and the DNA is isolated and subjected to density gradient centrifugation, a band is formed at the bottom of the centrifugation tube, and this is called as heavy band. When normal cells were isolated and DNA is subjected to centrifugation, a band is formed a little above the heavy band, and this is called as light band. So N15 containing a band will be heavier. This is heavy band and this is light band. Okay, so they have taken the E. coli cells and they have grown this E. coli cells in N15 containing medium for several generations, so that all the DNA will have N15 isotope. And then they have transferred or inoculated some of the cells in N14 medium. Okay, in the N14 medium, they have grown these cells for one generation. The generation time of E. coli is 20 minutes. So after 20 minutes, they have taken the cells and isolated the DNA and subjected to density gradient centrifugation. Now, there is a band which is neither light or heavy, but it is in between these two bands, like a hybrid band. So after first generation, a hybrid band was formed, and they have taken another sample after second generation, that is after another 20 minutes, that is 40 minutes. Now they have got a hybrid band and a light band. So two bands were formed, one is light and another one is hybrid. Then after third generation, that is another 60 minutes, they have taken the sample and they have isolated the DNA, and then they have subjected it to density gradient centrifugation. Now two bands were formed as usual, but the light band which is formed is thicker than the hybrid band. Okay, so this is the experiment which was done by Messelson and Stahl. Now we have the experimental results and three hypotheses, three modes of DNA replication which was hypothesized. Now the interpretation part contains, for example, if the conservative mode of DNA replication is true, it must be forming two bands. One is light band and one is heavy band. Why? Because according to conservative mode, the two strands of the parent DNA will be conserved in one of the daughter DNAs. So the daughter DNAs will have one daughter DNA will have both the strands N15, and the other daughter DNA will have both the strands N14. So N15 containing DNA will form a heavy band, N14 containing DNA will form a light band. So if conservative mode of DNA replication is true, it must be forming heavy and light bands, which is not the case with the experimental result, where only hybrid band is formed. So we can easily disprove the conservative mode of DNA replication after first generation of after first generation. Then coming to the semi-conservative mode. According to semi-conservative mode, the parent DNA, the two strands of the parent DNA will be separated, and the daughter DNAs will be formed on this parent DNA. So the daughter DNAs will have one N15 containing strand, and the other one is N14. That is one old strand and one new strand. So since since one DNA has both these N14 and N15 strands, it will be forming a hybrid band, which is true with the experimental results. Coming to the dispersive mode, according to dispersive mode, the daughter DNAs will have fragments of both parent and the new daughter DNAs. So the daughter DNA will have N15 isotope and also N14 isotope. So it will obviously form a hybrid band, which is also true with the experimental result. So by the end of the first generation, the conservative mode is disproved, but a semi-conservative and dispersive mode are proved. And after the second generation, According to the experimental result, two bands are formed. One is light band and then the one is hybrid band. In semi-conservative mode, this will be the parent strand, and the parent strand will be separated. One is N15 and another one is N14, and new strands are synthesized on these templates. So here two daughter DNAs are formed, and one daughter DNA has one strand is N15 and another one is N14. So it will form a hybrid band. The other daughter DNA has both strands are N14 containing isotope. So it will form a light band. So according to semi-conservative mode, after the second generation, one light band and one hybrid band must be formed, which is exactly true with the experimental result, and hence semi-conservative mode is proved. Whereas we should also look at dispersive mode. Dispersive mode will again form different fragments of DNA which contains both N15 and N14 isotopes even after the second generation. So dispersive mode must form a hybrid band even after second generation, which is not true with the experimental results. So dispersive mode is disproved. So by this interpretations of the experimental results, semi-conservative mode of DNA replication is proved, disproving the conservative and dispersive mode. So this is the experiment done by Messelson and Stahl. So in this video, we have seen that the DNA replication is an essential process for the cell survival. It follows certain basic rules, and the semi-conservative mode of DNA replication was proved by Messelson and Stahl. In their experiment, after one generation of growth in medium containing N14, and a density halfway between the densities of heavy DNA and the light DNA. This is called a hybrid. After two generations, half is hybrid and half is light. Conservative mode do not produce any hybrid, and hence it is disproved. In dispersive, they should observe a shift of DNA from heavy to light, which is not observed. Hence, it was successfully shown that DNA replication takes place by semi-conservative mode. Thank you.